So in terms of wavelengths, both visual and auditory sensations both occur in the form of waves. And so a wavelength is the length of a wave from one peak to the next, as you see in this example. So the wavelength is from here to here, from one peak to the next. The amplitude is the height of the wave measured from the highest and lowest points of the wave. So from the peak to the trough here. This is the measure of amplitude. Then the frequency is the number of waves that pass in a given time, and this is measured in hertz. So the frequency of this example would be 1, 2, 3. There were 3 waves in this given amount of time. And longer wavelengths have lower frequencies. As you see here, longer wavelengths have lower amounts of frequency, so there's only 3 waves here, whereas shorter wavelengths, as you see on the bottom, have more higher frequencies of waves occurring. This is the basics of waves and wavelengths. And moving on to light waves. Here is the light waveform spectrum, and the visual spectrum here is the portion of the larger electromagnetic spectrum that we can see. So this is the spectrum of light that we're able to see, and that's visible to us. Everything here is more microscopic, so we can't see atoms or molecules. This is more gamma ray, X-ray, and ultraviolet lights. And we can't necessarily see infrared, microwave, or radio waves. But we can see in this range of outcomes for light waves. And the electromagnetic spectrum encompasses all of the radiation that occurs in our environment. And this includes gamma rays, X-rays, ultraviolet, visible, infrared, microwaves, and radio waves. And the visible spectrum in humans is associated with wavelengths at the range of 380 to 740, which is, again, around this range here. And for humans, light wavelengths are associated with our perception of color. And within the visual spectrum, our experience of red is associated with longer wavelengths, where greens and yellows are more intermediate, and then blues and violets are shorter wavelengths. And then the brightness or intensity of color is affected by the amplitude. So the larger the amplitude is, the brighter the color appears, and the smaller the amplitude is, the more dull the color is. And then moving on to sound waves. The frequency of a sound wave is associated with our perception of that sound's pitch. A high-frequency sound wave are perceived as high-pitched sounds, so above here. And then low-frequency sound waves are perceived as low-pitched sounds, more on this decibel range. And the loudness of a cell, or of a sound, measured in decibels, is associated with the amplitude of the sound wave. So here you could see the measures of decibels. Soft music or a whisper is about 40 decibels. Intermediate of just hearing a car, or normal sound of just me speaking is around 60 to 80% decibels. And then as we move up in decibels, around 100, 120, or 140, we have the possibility of the risk of hearing loss. This can be an example of jet planes flying. Around 130 is our pain threshold, and around 140 decibels is really harmful for our ears. And higher amplitudes or higher decibels are associated with louder sounds, whereas lower amplitudes are associated with quieter sounds. Then moving on to vision, the anatomy of the visual system and of our eyes include the cornea, and this is the outside layer, a transparent layer covering the eye. We have our pupil, which is the small opening in the eye where light passes through. And in low light, our pupil dilates, or it expands. And in high light, our pupil constricts, or gets smaller. And this makes sense because in low lights, we need to expand the pupil in order to get more light to see, whereas in a lot of light or high light, our pupil gets smaller because we already have so much amount of light in which we can see. And the iris is the specific muscle that controls the opening and the closing of the pupil. And this is our colored part of the eye. And then the lens is a hard, transparent structure that reflects light to the fovea. And so light passes through our cornea, through our pupil, and refracts off of our lens to the fovea. And our fovea includes light and images, which are focused on the small indentation here of our eye, which contains photoreceptors of cones and rods. And this is also where our retina is, which is the back of the eye that receives light. And then cones, located in the retina and the fovea, are photoreceptors specialized to see color and specific details, whereas rods are photoreceptors that are specialized to see low light conditions and dull colors, and also peripheral vision. And so cones are really focused on this area right behind the lens for detailed and color, whereas the rods are mostly around our peripheral vision of our eyes. And then further, once light hits our optic nerve, this is where the cones and rods are connected to ganglion cells, which connect to the optic nerve at the back of the eye. And then in or near our optic nerve, we have our blind spot, which is a small point near the optic nerve where we are unable to obtain visual information. And then the optic chiasm includes how the optic nerve merges below the brain to the optic chiasm. So the optic nerve connects to the brain, to the optic chiasm. So visual information goes through our eyes, through the optic nerve, and then to the optic chiasm. And then visual information from our right eye is sent to our left side of the brain, whereas visual information from our left eye is sent through the eye, to the optic nerve, to the optic chiasm, which is then sent to the right side of our brain. And then how we see color, or how we perceive color, involves the trichromatic theory of color vision. And in this theory, all colors in the spectrum can be made with various combinations of red, green, and blue. And so with a combination of these three colors, we get the multiple different colors within our wavelength. And these three colors make up all combinations of the color spectrum. And we see spectrums of color through our cone receptors, and then the brain must interpret the sensitivity and the intensity of the cone receptors in order to interpret the kind of color that it's perceiving. And normal-sighted individuals have 3 different types of cones that mediate color vision. And according to the trichromatic theory, all you need to remember is that the combinations of red, green, and blue make up all the colors within the spectrum. And then there's also the opponent process theory, in which color is coded within our brain in opponent pairs or complementary colors. So black paired with white, yellow paired with blue, and green paired with red. And some cells in our visual system are excited by one of the opponent colors and then inhibited by the other color. And this is called the opponent process theory. And then a cell that was excited by, for example, green colored wavelengths would then be inhibited by the opposite or the complementary color of the red colored wavelengths. And so there's an excitation of one color, whereas the opposite color is inhibited. And for example, in this demonstration, I want you to stare at this image for about a minute, and you could pause the screen or the video in order to view that for a minute. And then once you view that image for a minute, I want you to stare at this little white dot for a minute, and then I want you to look at a white surface, so either your wall, a blank piece of paper, anything white, and you should see the opposite colors of this figure here. So take a moment to do that. Go ahead and pause the video now. Make sure you're staring at that white dot for a minute. All right, so hopefully you did that. And if you stared at a blank white piece of paper or a blank white wall, you'll see that this green color here turned red, and that this black color here turned white, and then the yellow color here turned blue. And this is because when you're taking in this visual information of all these colors, the opposite colors are the ones that are inhibited. So as you're looking at this picture here, maybe a flag, your cells in your visual system become excited by this green wavelength, this black wavelength, and this yellow wavelength. But only after you stop looking at this figure and look at a blank piece of paper that's white, you see the opposite color, because those cells and those colored wavelengths are inhibited, whereas the ones that you looked at here were excited. And this produces an after image. So the after image, you would see the red, the white, and the blue colors, as opposed to the initial colors that you see here. And then moving on to depth perception. This is how we perceive spatial relationships within our environment. And we use binocular cues to establish depth using both of our eyes. But we can also have different views of the world through binocular disparity. So if we take one hand and cover our eye, or if we take our fist out and we put our thumb up, and with both eyes we could see the thumb, but if we close one eye or cover one eye, and do that successfully with both eyes, so first we cover our right eye, and then we cover our left eye, we should see the thumb move over and see that binocular disparity. And then we also have monocular cues, which establish depth with only one eye. And so if we only look at one eye, we might lose part of our vision. And then linear perspective is when we see depth with parallel lines establishing a sort of direction. As you see here with this photo, we see parallel lines going forward into the distance, and this creates a depth perception of a linear perspective moving through the image.